Welcome to AI for Genomics Data Analysis from 𝗿𝗲 Academy. In this session, we look at how machine learning reads the genome, from dimensionality reduction all the way to foundation models for single-cell and spatial biology. Modern genomics generates data at a scale no human can read by hand. A single brain atlas experiment profiles millions of cells, measures tens of thousands of genes per cell, and can map billions of spatial transcripts. Distinguishing hundreds of neuronal and glial cell types is a needle in a haystack problem that spreadsheets cannot solve. Traditional analysis hits a wall for 4 reasons. There are far too many dimensions to plot. The biological signal is buried in technical noise and dropouts. Samples from different days or platforms never quite line up, and genes act in combinatorial, nonlinear programs that simple models cannot capture. 5 families of AI power modern genomics. Unsupervised learning finds structure with no labels. Supervised learning classifies cells from labeled reference atlases. Deep learning models noise and nonlinearity end-to-end. Foundation models are pre-trained on millions of cells, and generative models let us simulate states we never measured. It helps to separate 2 paradigms. Unsupervised methods discover structure on their own, clustering neurons into candidate types, and building maps with UMAP. Supervised methods learn from labeled references, annotating new cells against a brain atlas and predicting their state. Dimensionality reduction is the workhorse. We start from 30,000 gene dimensions, compress them with principal component analysis, build a graph of similar cells, and lay it out as a 2-dimensional UMAP you can actually see. Autoencoders push this further, learning a compact latent space that denoises the data while preserving biology. Clustering turns a cloud of cells into named populations. Graph-based methods like label group cells by shared neighbors. The resolution setting matters. Too low and rare interneurons vanish. Too high and one-type splinters into noise. Clusters become real biology when their marker genes match known signatures. The payoff is hundreds of brain cell types resolved from a single dataset. Variational autoencoders learn the biology beneath the noise. Each cell is squeezed through a narrow neural bottleneck and then reconstructed. To do that well, the model must learn a compact latent code that captures real variation and discards technical noise. It models raw counts directly, corrects batch effects, and scales to millions of cells. Batch integration makes biology line up across samples. Before correction, cells split by sample instead of by type, so 2 batches of the same cortical neurons sit in separate islands. After integration with a method like harmony, they overlap and cluster by true identity. Getting this right is the difference between a real discovery and a batch artifact. AI can put names on cells far faster than manual gating. Reference mapping transfers labels from a brain atlas. Classifier models score every cell in seconds. Language models can even suggest identities from marker lists. But the reliable pattern is human in the loop. The AI proposes, and the biologist verifies. We can also infer the wiring diagram of gene regulation. Cell identity is set by transcription factors switching genes on and off. AI reconstructs these networks from data, co-expression links, regulins that group a factor with its targets, and enhancer to gene connections revealed by 𝗿𝗲 data. Deep learning can read the genome letter by letter. Convolutional and transformer networks take raw DNA as input and learn regulatory grammar that no one wrote down. They predict which chromatin will be open, score enhancers and their targets, and estimate how a single base change disrupts regulation. Foundation models are the current frontier. Trained on tens of millions of cells, they learn a general language of gene expression, which you then fine-tune for your own task with far less data. They help with label transfer, perturbation prediction, integration, and network inference. But they are not magic. They inherit the biases of their training data and still need validation. Spatial methods add location, where each cell sits in the tissue. AI deconvolves which cell types make up each spot, learns how neighboring cells influence each other across a cortical layer, and carves tissue into spatial domains that share an expression profile. Multirome analysis joins modalities. Expression tells you which genes are active, chromatin accessibility tells you which regions are open, and protein markers add another view. Deep models fuse these into one representation, so a neuron is defined by its expression and its regulatory state together. Generative models let us run experiments in silico. We can knock out a transcription factor on the computer, predict the resulting shift in expression, and use that to prioritize which experiments are worth running at the bench. It is a way to ask what if before committing reagents. A prediction you cannot explain is a hypothesis, not an answer. Feature attribution shows which genes drove a call. Attention and saliency reveal where a sequence model looks. Calibration tells you when the model is unsure. And the final check is always biological validation, not the loss curve. Be aware of 4 common failure modes. A model can erase a batch and hide real biology, or invent structure that is pure artifact. Data leakage between train and test inflates accuracy. Rare cell types are easy to overfit, and confident language model outputs can name a type that no marker supports. Always verify. A responsible workflow ties this together. First, quality control. Filter, denoise, and remove doublets. Second, represent. Reduce dimensions and integrate batches. Third, model, cluster, annotate, and infer networks. And fourth, validate. Check calibration, interpret features, and confirm at the bench. To sum up, AI is how we read genomics at scale. We reduce and integrate, we discover and annotate, and we predict and simulate beyond what we measured, but we always validate. Thanks for learning with 𝗵𝗲 Academy. Find more at 𝗵𝗲_Academy.com.